Hello everybody, welcome to MDC Connect's webinar series. I'm Sarah Brockbank, I'm a lead scientist at Medicine Discovery Catapult, and I'm hosting the session today with my colleague Dan Bond. This is now the 7th in a series of weekly webinars that run every Wednesday at 2 o'clock. So this is a series of 9 informative drug discovery sessions, each delivered by experts at Medicines Discovery Catapult and also our CRO network partners. And each of the sessions is focused on an aspect of preclinical drug discovery. And we started with the selection of the right target and identifying the hit. We thought about optimizing the compound and a focus on the chemistry involved. Last week we introduced PK/PD, and today we'll be talking about target validation, and we'll finish the series by defining safety and biomarker strategies. So, as I said, in this session we're thinking about target validation and efficacy. And I'd like to welcome 3 speakers, or at least I hope I'm welcoming 3 speakers, because we're just getting them in as we started. So firstly, Matt Burnham, who's a lead scientist here at Medicines Discovery Catapult. Matt is going to highlight the importance of target engagement and describe the methods used to confirm it. Then we've got Amanda Woodrooffe, who's a Senior Vice President at Precision for Medicine, and she is going to go native. So Amanda's going to describe the merits of using human primary cells. And finally, Lorraine Mooney, Study Director at ONCOLOGY. And Lorraine will talk about the use of in vivo models, the advantages, disadvantages, and the challenges of demonstrating efficacy in preclinical models. So we will take questions at the end of each presentation. And if you wish to ask a question, you can type these at any time into the Q&A box at the bottom of the screen, not the chat box. And if you put your questions into there at any time through the course of the presentation, the speaker will pick them up at the end.
Thank you, Sarah. Hopefully you can hear me and you can see my slides.
Yep.
And today I'll be talking about target engagement. Particularly in cellular assays, and why that particularly is important, and what it means, and also I could be giving you a few examples. So it's important, target engagement is important because it allows you to test your biological hypothesis. That drugging a particular target is going to lead to a particular therapeutic effect. And to be able to test, is that idea right or wrong? So it's a key piece of the whole target validation process that we've talked about today. And we know, or we now know from various published studies, that lack of efficacy has been the leading cause of drug failure in clinical trials. And if you can't show whether your intended target has been modulated by your compound, which is proof of mechanism, how do you know whether that lack of efficacy is due either to the drug not hitting the target, or alternatively, and really completely opposite result, that the drug does hit the target, but that target does not modify the disease? One's devalidation of the molecule, and the other is devalidation of the target. And it's very different outcomes and have very different next steps. So if we look at an in vitro cascade, like in an early discovery cascade, we have a hit or a lead molecule that we're bringing forward to a cellular assay, a phenotypic assay that represents some aspect of the disease biology. We might see either, well, if we're unfortunate, we might see no effect of that compound. We'd immediately start to wonder questions such as, did the compound actually get inside the cells? Or if it does, is the target in this context somehow different than in previous assays? And do I need a better molecule to see activity. But on the other hand, if you do see a cellular response, how can you be confident? How can you really build your confidence that that response is due to activity at your intended target? It's not an off-target effect, and it's not a non-specific effect. And also, the cellular potency, you want to understand that in terms of the other potencies that you've measured in other contexts. And all of these questions are part of target engagement. So target engagement, it can be broken down into a series of 3 steps, really. The first is, does my lead molecule reach its intended site of action? This could be PK type questions about crossing the blood-brain barrier. It could be questions whether your target is expressed on the cell surface or intracellular. If it is inside the cells, your cellular permeability, that could be very different in a bacterial cell or a human cell. Or a tumor that expresses efflux pumps and it removes the compound from the site of action. Second question is, does my ligand interact with its target in a way that's intended, with the mechanism that's intended? And if your target is a well-characterized class of targets, it may be that it's fairly straightforward to do that, and there are well-established methods. Alternatively, if your target is a highly novel target, it can be extremely difficult, actually, to demonstrate target engagement. If you don't have probe compounds, or protein is difficult to purify and handle, it can actually be considerable effort to try to demonstrate that. And thirdly, what are the downstream consequences of your ligand-target interaction? So here we're looking for proximal markers that reflect both the interaction of that ligand with its target and report on that, but also markers which can demonstrate that the disease pathway has been modulated. And if you can get some idea of the amount of modulation of the pathway that's needed to modulate the disease, then you can try and tie that to the amount of activity at your target, the amount of target occupancy you require for the desired effect. So, for an example here, hits are often identified in a screen that uses a purified protein as a target. And it might be very amenable to use that purified protein in a biochemical or biophysical characterization of its mechanism and its binding characteristics. But that purified protein is unlikely really to represent the target as it will be in its cell environment. It might only be a protein domain instead of a full-length protein, or even a full-length protein isn't going to have all the modification, all the phosphorylations and the modifications that a cellular protein would have, and it won't have the proteins that it interacts with, and it have the cofactors, and potentially a whole entire macromolecular complex that could be surrounding your target. And the protein target in a cell environment, it's not a static environment. That cellular environment changes over time, and even the cellular microenvironment can influence that. So this is where cellular target engagement approaches are really so important, because they try to model that complexity in that dynamic environment that represents the cell environment. And I think actually the earlier talks really have really given a good overview of that the different range of cellular and in vivo models that we can use to try and recreate the most accurate and translatable environment as possible. But for cellular target engagement, you have it'd be a target that can be expressed on the cell surface, a receptor such as a GPCR, and there's some well-established methods, competition binding, can characterize that ligand-target interaction. You also have the advantage that your assay reagents don't need to be cell permeable in order to perform that analysis. But if your target is intracellular, there's actually quite a limited number of approaches that you can apply in that situation. And they actually vary quite a bit in the types of data and the complexity to set them up. So this is the next couple of slides will focus on, this intracellular target engagement approaches. And they can be sort of categorized broadly into 3 categories. You can start with the area in red there, the label-free approaches. So this is where you don't need a modified ligand or a modified target. And CETSA is a good example of this sort of an assay. And that's really the advantage here, is that you don't need that modified reagent. You can just get going with your ligand, your compound of interest and appropriate endpoints and get going. On the other hand, you might have an approach such as BRET, which requires both modification of ligand to form a tracer molecule and also modification of the target itself. But the advantage here, the trade-off here, is that you get much more detailed characterization data, so your characterization of that ligand-target interaction in the intracellular environment, much more detailed. And then there's a range of other approaches here shown in yellow, which rely just on a modified ligand or tracer. Really nice recent one from an oncology paper is actually to use a PROTAC approach. So converting your ligand of interest into a PROTAC and then demonstrating target engagement by the PROTAC-induced degradation of that target, selective degradation of just your target. However, I think these modified ligand approaches, especially the chemical proteome affinity-based, photoaffinity-based approaches, really require detailed SAR understanding of your ligand and the difficult approaches to achieve in practice. So I'm going to focus on 2 approaches, the CETSA and the BRET approach, and give a little bit more methodological detail. So BRET which is similar to FRET, but is the bioluminescent resonance energy transfer process. That's actually a ligand displacement assay that you can perform inside live cells. You need the tracer with a fluorophore and the target to have a luciferase tag. And you actually have a very, very specific signal in that situation, because your target is generating the light signal that gets transferred to your tracer in the BRET reaction. And then you can follow binding of your compound of interest as it displaces that tracer and reduction in the BRET signal. And the data panels on the right there, they're taken from the original publication. And they focused on a series of histone deacetylase inhibitors tested in a panel of HDAC isosomes. And as well as being able to characterize the binding of these compounds against those isosomes in their cellular environment, it also showed factors such as prodrug, which only became active in the cellular environment. They're able to assay that behavior. But I think one of the sort of main attractions of the BRET approach is that it offers ability to measure residence times, intracellular residence times. This is one of the parameters that's seen as there's a lot of interest in that area for improving the drug safety and efficacy profiles. You can see the ability to measure that there is given in that panel, and the differences in the unbinding kinetics of the different compounds. And it was possible actually to explain known cellular activities and very different cellular activities on the basis of those very different residence times. BRET is also, it's well served for the kinase field. There's range of tracer molecules are established, and 6 tracer molecules actually cover the majority of the known kinases. And in fact, the approach has also been demonstrated in xenograft preclinical models now as well. The Cellular Thermal Shift Assay, which is CETSA, and is a label-free approach. And what it relies on is that binding of a ligand to its target stabilizes that protein structure and then shifts the temperature required to denature it to a higher temperature. And that generates what's known as a melt curve, and you can see that shift in a melt curve. And the data in the middle panel there is from the original Karolinska Institute publication. And they've shown there the PARP enzyme target with a compound that does bind to the target, in this case olaparib, and that's in blue. You can see the shift there to a higher temperature compared to a compound which doesn't bind the target, iniparib, in green. I'll actually come back to these compounds on my last slide. The panel on the right is an alpha-lyser higher throughput endpoint overlaid on top of the basic CETSA approach. It gives you higher throughput. What these higher throughput endpoints have shown actually, quite interesting for the whole concept, is that there's a subset of compounds that bind as high affinity binders to a target. With just the particular arrangement of that interaction of that particular ligand with the target doesn't stabilize the target structure. And you don't see a CETSA shift for that binder. So there's a category, a subset of compounds which are CETSA false negative compounds and can't be detected by this approach. But generally, the approach is actually very flexible, and there's a number of wide range really of endpoints that can be overlaid, and a number of them that are suitable for the cellular applications, so as proteomic readouts, imaging readouts, and even ex vivo analysis of samples. Now on to proximal markers, and just to highlight a few aspects of this. What you're looking for with a proximal marker is something that's well validated and specific. It's well validated to be specific to that interaction between your ligand and that intended target. You want these markers of that ligand-target interaction and also potentially of modulation of the disease pathway to be detectable in your cellular assay to start with, but ideally they'd be able to translate to tissue studies and other samples, so that as your project moves forward, you can bring those same markers forward as validated markers. There's a number of approaches, or a number of really aspects of the target biology that could form proximal markers. In fact, there's a vast number of aspects of target biology. Highlighted just a few in those panels on the right there. But what I'd like to actually highlight are the technological approaches, particularly some which are available in the Medicines Discovery Catapult, and they're particularly geared towards these analysis of proximal markers. So nanostring and the digital spatial profiler are about highly multiplex detection in tissue sections and other samples. The single molecule array Simoa platform is geared towards extremely high sensitivity for detection of peptides and proteins. And microscopy platforms across a range of modalities for high content biology, but also protein scale super resolution imaging, and also tissue imaging with 2-photon. So I mentioned that I would come back to iniparib, and it's really the sort of story we want to avoid. In fact, it's a compound that was one of the early targeted therapies, and it was brought forward to phase 3 trials, despite having no demonstration of proof of mechanism. And it failed in those phase 3 trials. And retrospectively, looking back afterwards, they basically determined that the nipirib didn't bind, was unlikely to bind the PARP enzyme target at all. And if you remember from those earlier data slides, effectively what was a fairly simple Western blot experiment in vitro in cells could have demonstrated that lack of target engagement way before the project got to phase 3. So just to summarize, as target engagement, we're looking to show that ligand reaches its site of action, confirm that ligand-target interaction and its mechanism, and also identify and measure proximal markers that show report on that interaction, modulation of the disease pathway, and ultimately that the process is about validation of both the lead and the target. And thank you, Sarah.
I'm going to kick off by just providing some examples of the use of primary cells to support target validation and really try and convince you why you should consider going native. So in terms of in terms of presentation as an overview, I'm going to just talk a little bit about target validation and where cellular models fit within that. Talk a bit about primary cells. They're like everything, it's a model, there's good, there's bad, there's opportunity. And then I'm just going to present 3 case studies really that demonstrate why it could be important to go native with your models, talk about the importance of assay development, characterisation and appropriate level of validation, so you end up with a fit for purpose assay. And then the final case study, I'm going to just give an example of an assay that was developed primarily to support clinical work, but is also being used in discovery. And it's a quite nice example of how assays can be used from discovery through to clinical to support that translational piece. Okay, so to start with then, just in terms of target validation, obviously there is still a challenge for the industry in terms of clinical attrition, so the loss of compounds in clinical development. And the primary reason, unfortunately, that is the case still remains a lack of good efficacy in the clinic. And so ways to help to mitigate this is to enable a better and a thorough understanding of complex biology, complex diseases in particular, and the target biology associated with those, and also come up with better human and, importantly, clinically predictive models and biomarkers. So the failure rate during clinical development is often attributed to the fact that there's been inadequate preclinical target validation, or that you don't have something that is actually suitable for clinical translation. So I'm a firm believer that the development and application of really relevant preclinical cell-based models can help. And primary cells have the advantage that they are native, and they're kind of the most native biology that you can access preclinically. So as with anything, there are lots of important considerations that you need to think about before you start. So first and foremost, what is the biological question that you are looking to address or understand? What data do you need to generate? What relevant data is going to enable you to take your program forward? How are you going to go about characterising the relevant biology in your model? What type of model is going to be most fit for purpose? How many compounds have you got? How many donors do you need? What's the impact of donor to donor variability? So you need to think about what type of model you actually want to develop first of all. And I think also linked to that is the complexity of your disease. So what is involved in the biology of that disease, or the pathology of that disease? Are there different cells involved? You need to make sure that your model represents all of those. So there's a lot of things to think about before you even make a start. So just some images on the left-hand side, just to show you various human primary cells in different culture formats. I'm just going to run through some disadvantages. I'm going to start with the disadvantages, and then hopefully by the time we get to the advantages, opportunities, and positioning, you'll be slightly convinced. So in terms of disadvantages of using primary cells, the first and foremost is often cited as availability and sourcing. We just can't access them regularly enough. Scale is another issue. Many primary cells don't proliferate. They're terminally differentiated, so you can't bulk them up to get high throughput screens. They all come from individual donors. We're all different. We're an outbred population, so my hepatocytes are going to be very different to somebody else's hepatocytes, for example. And the challenges of actually being able to really well maintain the cellular phenotypes that you need when primary cells are in culture. So they do have some challenges, absolutely have some challenges. However, as I touched on just now, they are, from an advantages point of view, the most in vivo-like that you're going to get. And obviously, if you're able to use human primary cells preclinically, then that's going to give you the most native clinical model. The targets within the cells are native. You haven't got sort of overexpressed recombinants or targets that have been influenced by virtue of a cell line, for example, being immortalized. I said a disadvantage was inter-individual variation, but actually that could be an opportunity as well, or an advantage. You know, if you actually want to understand what you're likely to see when you go into the clinic by looking across a panel of different donors, then that can actually be an advantage to using native primary cells. And to address the challenge of maintaining cellular phenotype in culture, there's lots and lots of different culture formats and media out there that enable really good human primary cell models, but human primary cell models too, to be developed and maintained. In terms of opportunities, there are opportunities to do high volume isolations. I'm thinking here particularly of things like PBMCs, which can now be isolated in huge number from Luke-Pack draws, which you can get from commercial donor centres. The ability to cryopreserve cells, you know, does enable you to do a big prep, cryopreserve them all down and keep going back to the same donor all the time. And also pooling donors, and you see this a lot with in the hepatocyte world in particular, where different donors can be pooled together to give you a big stock of cells for your cell-based assays, and also helps to essentially kind of prevent some of that inter-individual variation. You're creating sort of a generic pool of cells from multiple individuals. In terms of positioning, typically primary cell-based assays are positioned at that sort of tertiary screening stage, candidate selection. So typically the type of work that we do, we're looking at sort of tens of compounds and not hundreds and thousands of compounds. I mentioned PBMCs earlier on, which can be isolated in very high numbers, and so they can be used for higher throughput screens. And again, I think when you're thinking about how your assay is going to position, thinking about the clinical translation element of that and biomarkers that you might want to carry through all the way into clinical development is definitely worth having to think about in terms of where you're going to put that assay in your cascade. So the first case study that I'm just going to run through is really a kind of almost a back to basics, really. It's really an example of making sure that your model is telling you what you need it to tell you. And this is an example from data that was generated quite a few years ago now. And it's all about understanding whether you've got the right biology. So basically, this was a comparison between a native lens epithelial cell line, so that's NLEC, and a lens epithelial cell line, which is HLEB3. And these two cell types were used to look at agonist-induced calcium release. And when you look at the increase in intracellular free calcium in response to acetylcholine in these two different cell types, they look pretty similar. So you can see the graph on the left-hand side, you know, they're fairly comparable. You've got pretty consistent stimulation in both the cell line and in the native cells. So at face value, that looks all right, looks pretty good. However, when you then start to antagonize those responses that you see in either the native cells or the cell line, when you use different antagonists, and in this case we were actually trying to interrogate the muscarinic and muscarinic receptors within these cell lines, because they've been implicated in some of the issues around use of acetylcholinesterases in glaucoma. So we were looking at an M1 antagonist, muscarinic receptor 1 antagonist, and also a muscarinic receptor 3 antagonist. So when you add your antagonists in and you look at the pharmacology, then actually you're seeing very different responses. So in the native cells, the M1 is a lot more potent, and in the cell line, the M3 is a lot more potent. So we then generated some qRT-PCR data to have a look at actually what receptors are expressed in these different cell lines. And sure enough, as is supported by the pharmacology data, the native lens epithelial cells primarily express the muscarinic 1 receptor, and the cell line primarily expresses muscarinic receptor 3. So this is really just a fairly simple example of make sure that you understand what your model is going to use, is going to represent. So for us, this was the primary cell model was the preferred model because the cell line was not representing the correct biology. Okay, I'm going to come on and talk a little bit now about assay development and the importance of characterizing the model. So I'm going to talk about a 3D model that we've developed here in our labs in Royston in the UK. And it's a model that is representative of the pancreatic islet. And it was a model that was developed to support diabetes research. So the pancreatic islet is itself a 3D model. So if you see the panel, the top of the 2 panels on the very left-hand side, they are native islets. Which have been sort of freshly isolated from cadaver pancreas. So they are considered definitely the gold standard when it comes to understanding islet function. However, they're incredibly hard to work with. So essentially what we tried to do and what we achieved is we created a model of these native islets by putting them into a controlled sort of 3-dimensional co-culture model. And first and foremost, what we needed to look at was whether that model actually represented and recapitulated the native islet morphology that you see, and whether we could keep that going in prolonged culture. So the panels that you see in the center of the screen at the bottom there are really looking at pancreatic islets in situ. So this is in sections of frozen pancreas. So this is homing in on an islet in a piece of tissue. And we're looking at markers for the alpha, beta, and delta cells that are present within the pancreatic islet itself. And so the first 2 boxes at the top show co-expression of insulin and proinsulin. So you get this kind of yellowy overlapping color here because the 2 are both expressed in beta cells, so they're co-localized. Then as we move across to the right-hand side on the top, we've got insulin and glucagon. So insulin is a marker for the beta cells and glucagon for the alpha cells. And then the final panel over on the right-hand side at the top there is insulin and somatostatin. So again, insulin for the beta cells, somatostatin for the delta cells. So we can actually see that the 3 cell types in the native pancreatic islet are all there, and they're fairly mixed in terms of their distribution. When we then look at the organ dot cultures that we've created using native islets, so we essentially take the islets apart and put them back together under controlled conditions, we can see that the model itself is recapitulating the morphology of the native islets in situ. So in terms of the list at the top there, so the model we've demonstrated it recapitulates the morphology, and that's obviously a good starting point for us for the model development and characterization. The next is to make sure, and it's unfortunate that Matt didn't go first because I was going to refer back to Matt's presentation about making sure that the targets are there so you can engage, your drug is engaging with the right target. So making sure the target is there. So we did some, again, QRT-PCR work to make sure that the key pharmacological targets that are being looked at for the type 2 diabetes, the one that I'm going to present a case study on in a moment is GPR40, so remember that one for the next slide, to make sure that they are expressed. And so the data here just show that we've got native islets. This is delta CT, so the lower the number, the higher the expression. So you can see there's stack loads of insulin, which is these white bars that go downwards in both the native islets, but also in our islet organ dot cultures at both day 12 and day 20. So that's giving us the longevity in the model that we were looking for. And then there's a whole range of different targets that are involved in islet biology here. And as you can see, the hashed, the sort of horizontal hash lines, which is 1, 2, 3, 4, 5 bars in, that's GPR40, which is actually our target of interest for the data that I'm going to show you in a moment. So making sure your targets are there and they are maintained in your culture environment is important. And then make sure that they actually do what you need them to do. So this was a model where we were looking at glucose-stimulated insulin secretion. And we're looking at response to insulin and to standard secretagogues. And secretagogues are compounds that potentiate the insulin secretion. So the data at the bottom just show example data from 5 donors where we're able to see an increase in insulin secretion from our islet model with increasing concentrations of glucose to stimulate that. And then in the presence of extendin 4, which is Byetta, which is one of the marketed type 2 diabetes drugs, we're actually seeing a potentiation of that insulin secretion. So, you know, the compound is driving the beta cells to pump out more insulin to correct the blood glucose. Reliable and robust methods as well, really important. We've now generated data from over 80 individual donors of pancreatic islets that we've dissociated and put into our model. And we do get really robust and reliable insulin secretion from every donor. I think maybe one we've had that didn't work terribly well. So that's a pretty good hit rate for a primary cell-based assay. So with this assay format in mind, what I'm going to do, I'm just going to take you through to the second case study. And this is, I kind of cheated a little bit here because I've got a little bit on efficacy, but I've also introduced an aspect of safety as well, because I think it's really important to remember that the two go quite closely together. And I know we're going to be hearing about safety, I think, at the next webinar. So this really is a case study about getting that balance right. So you'll remember our friend GPR40 from the previous slide. This is one of the targets that is being pursued, I guess is the right word, as a treatment for diabetes. So Takeda had developed a potent and selective GPR40 agonist. And the theory behind that is that they are supposed to be safe and effective as an anti-diabetic drug, and not cause some of the issues that come up with other mechanisms causing hypoglycemia. So TAK-875 was their compound of choice, and certainly in our model, when we looked at this compound in our model, so if we look down at the bottom left-hand side, there's two different donors of islets here, and what we've got is increasing concentrations of the Takeda compound at 6.7 millimolar glucose and 16.7 millimolar glucose. So we can see that that we're getting potentiation, we're getting a nice concentration dependent increase in insulin secretion. So in both donors, so the drug was pretty effective at potentiating insulin secretion. So that's great. And if you took that data in isolation, you'd be thinking, great, this is brilliant. My compound's amazing. It does everything I need it to do. Unfortunately, Takeda, the drug was withdrawn from phase 3 clinical development, and that was due to drug-induced liver injury. So we took that information and we did some retrospective analysis in BioIVT's cholestatic drug-induced liver injury model. And this is essentially, it's another human primary cell-based model. It's sandwich culture of hepatocytes. And when we looked at the compound in this model, so it's basically a sensitization type model. So actually what we're seeing is that with the Takeda compound here, which is, if you look at the, I don't know if you can see my pointer, you probably can't see my pointer. So if you look at the two graphs on the bottom right hand side, the Takeda data, Takeda compound data are both on the right hand sides of those graphs. And when we get up to 100 micromolar, in the C-DILI model, we are actually seeing quite significant toxicity that's happening with the compound. So really what I wanted to do was try to give you an example of where actually using, in this case, two primary cellular models in combination allows you to both sort of validate the target and look at your drug effect, but also, in the same breath almost understand the potential for any safety liability. The last example I just wanted to give was an example where we were asked to generate or develop a model for screening anti-TNF alpha biosimilar.
Amanda, do you want to start wrapping up?
Yep. So basically, this was something that was developed for clinical application, really, because we were looking to support data for a marketing authorisation application. So we took it through a full assay development, which involved looking at standard inhibitors, titrating with stimulus, looking at sentinel markers, all the things that you would want to incorporate. We then took this assay through full US Pharmacopoeia guidance for cell-based assay validation, which included actually looking at different formulation effects, which I think Lorraine's going to mention later on. And then we were able to generate data on a whole series of biosimilars and look at their relative potency versus Humira, which was the marketed compound that's coming off patent. This is a nice example because we are actually also using the exact same assay to screen small molecules in early discovery as well. So this is an example of an assay that you could take through from early discovery right the way through to support some of your clinical data. So to summarize, really just keep thinking about clinical translation. It is possible to model native biology at native receptors in complex systems. You can do it. Think about where your assay needs to be, the format, How complex does it need to be? Different cell types? What throughput do you want it to be? Can you get access to the materials you need? Characterize it as fit for purpose. It's a model. It's never going to be perfect, but it needs to be able to generate the data that you need to progress your program. So make sure that your model delivers. And then look at the level of validation that you actually need. So multiplex endpoints could be a good start, and then you triage down to something that's easier to manage, clinically relevant, or could potentially be a clinical biomarker. And last but not least, I would always advocate to consider the bigger picture, and that's looking not just at efficacy but also at safety in these primary cell-based models. And with that, I'm going to finish up and leave you with a smiling cell.
That's lovely, lovely. Thanks, Amanda.
Okay, good afternoon everyone, and thank you for the opportunity to talk today. So the title of my talk is the use of preclinical models to deliver proof of concept efficacy. So this is an outline of what I'll cover. So I'll briefly tell you who we are and what we do, and talk about proof of concept and things that you would want to consider before moving into in vivo oncology models. And I'll then present two short case studies, one on FIBROBLAST growth factor receptor inhibitor and one on a PI3 kinase inhibitor, and almost revisiting mechanism of action in immuno-oncology. And then lastly, I'll summarise. So we are Alderley Oncology, and as the name suggests, we are based at Alderley Park. We are a CRO that provides in vivo pharmacology services, and we've been established for just over 2 years, and we are all ex-large pharma. So on this slide is an image of a typical oncology study and the type of work that we do, where we can take tumour cell lines, implant them either subcutaneously or orthotopically into immune competent or immune compromised mice, allow engraftment, and then randomise them onto study and treat with therapeutic agent and look at tumour growth over time. And you can see on the right hand side there a tumour growth inhibition curve. And we can also take samples from these studies to look at pharmacodynamic biomarkers. So we are a fee-for-service, work on a fee-for-service basis, and we have experience working with a range of therapeutic modalities. So I guess over the last 6 weeks, you've heard from various experts from talking about target identification through to lead optimization. And last week we discussed the importance of PK/PD studies and using that to help guide your dose and schedule for proof of concept efficacy studies. So in this scheme here are the types of studies that we can do and highlight the type of work that has enabling work that has to be done before you get to that proof of concept in vivo. So we can do these with a lot of these models with commercially available cell lines, or we can actually do bespoke model development for you. So in terms of if you look at your pharmacodynamic studies, we can look at target engagement in normal and in tumour tissue. Often in oncology, efficacy studies can run between 2 and 4 weeks. So we need to understand your therapeutic PK, not just from an acute dose, but also on chronic dosing. And we need to understand that your agent at the dosing schedule that you've decided on based on your PD studies is actually tolerated for that schedule before you proceed to your in vivo proof of concept efficacy study. So I guess in terms of target validation, this is absolutely key to success and is obviously something that continues throughout the lifetime of a project. And as you, I guess, increase the biological complexity of the system, looking at cell lines, primary cells to organoids, then going in vivo and clinical, it becomes much more challenging as that biological system is more complex. But at each step, as you achieve positive data, you're validating your target and decreasing the risk of your project. So what does it take to get a molecule ready for in vivo? So this is an example of a drug discovery cascade for a small molecule. If you look at, I guess, the top half of the cascade highlights the in vitro pharmacology, and the bottom half highlights the type of assays that are run in the in vivo pharmacology. And if you look at the right-hand side, you can see numbers which are associated with the number of compounds that you put through the cascade. So obviously, as you go through each of the different assays, you're filtering the compounds and only selecting those that have the desired properties to take through to the in vivo studies. And as you increase the number of assays and increase your data sets, you're constantly building that platform of evidence for your target in that particular disease setting. So from an oncology perspective, there are a number of models, murine models, that are available to evaluate your novel therapeutic treatment. And this slide highlights those models. The model choice is dictated by your target, can be guided by your in vitro cell line data, and obviously the hypothesis that you're testing. So more commonly used models are SYNGENEIC models and human cell line derived models. SYNGENEIC models use mouse cell lines that are grown in immune competent animals and are a platform for testing immune therapy agents. Similarly, genetically modified models, which tumour development is driven by a genetic alteration, can also be used for immune therapy. The disadvantage of these models is that the penetrance of the tumour phenotype and the latency of the development of the tumours. Human cell line derived models are using human cell lines grown in immune compromised animals, but are workhorse models in oncology drug discovery. And then the last two that I've highlighted here, patient-derived explant models and humanised models, are increasingly being used within oncology drug discovery. Patient-derived explant models take a sample directly from the patient and grow this in immune-compromised animals. And they produce the complexity, I guess, of the human disease, maintain that genomic heterogeneity. Humanised mouse models, I guess, are still under development, a really valuable tool, and I guess I'll mention them later. So in terms of what you need before embarking on that proof of concept in vivo study, there are some things to consider, and I guess I just wanted to pull out some things to think about. So for experimental design, so the suitable tool molecule for testing, you really want to be confident that you have the potency and selectivity and good PK properties before you go in vivo. You want to understand the PK of your compound in terms of that dose and schedule, both from an acute dose and also on chronic dosing. You would ideally like to have a good an understanding of your PK-PD relationship, but that's not always known at the start of your in vivo cascades. Formulation, you want to have a stable formulation for chronic dosing. You want to start to think about controls, what vehicles you need to include, and then positive controls. Do you want to look at your agent against standard of care in a particular disease type within oncology? And also monotherapy or combination. Is your agent going to go into the clinic as a monotherapy, or actually will it be dosed in combination? And that's something you really need to start thinking about early on. And also for many of the studies that you do, you want to get the most out of these studies, so making sure you take as many samples as possible to generate, understand your PK, tumour for biomarker assessment, and indeed other tissues to look at any other kind of normal tissue biomarkers. So the model choice, and it's about thinking about what your mode of action is. Making sure that your models are well characterised. So for example here I've highlighted immune phenotype of SYNGENEIC models. So in the pie charts are 3 different SYNGENEIC models, and each colour within that pie chart represents an immune cell type, and you can see between the 3 models they're quite different. So you want to make sure that you select the model that has your desired cell type of interest expressed within the tumour microenvironment. For targeted therapy, cell line derived xenograft models can be used. If a cell line isn't available, you can use patient derived xenograft models. And for immune therapy, SYNGENEIC models, also genetically modified models and humanised models are commonly used. But I think overlaid on top of all this, it is about understanding your model and the utility of the model. They all have advantages and disadvantages, and it's about being able to interpret and use the data that you generate from your studies in the right way to be able to drive your project forward. So I'll move on to the case study. So the first one is fibroblast growth factor receptor. This pathway is quite a complex pathway. There are 22 ligands and 4 receptors, and they are key in multiple biological processes, including tissue repair, angiogenesis, and embryonic development. And this axis has been implicated in tumorigenesis and indeed chemoresistance. On the right-hand side here, you can see some molecular profiling that's been done looking at the receptors and showing that across multiple different cancer types, that there is a molecular aberration of different receptors in these cancer types. So the concept was that by inhibiting this pathway, you had opportunity to impact multiple different cancer types. So AZD4547 is a potent and selective inhibitor of FGFR 1, 2 and 3, and weakly inhibits FGFR4. Now, from a huge amount of in vitro data, we identified tumour cell lines with molecular aberrations in FGFR that were sensitive to 4547, both from in terms of proliferation, but also target engagement, showing impact on phosphorylation of the receptor and downstream signalling. So we wanted to test that in vivo and determine whether we could see similar effects in vivo. So we selected some of the cell lines from the in vitro screen, and we were able to grow these in vivo in immune compromised animals. So one was KMS11, which is a multiple myeloma line with an FGFR3 translocation, KG1A, which is a myeloproliferative disease and has an FGFR1 translocation. And we also included CALO6, which is wild type for FGFR. And you can see from the curves along, the curves along the bottom of the slide that we've seen significant effect on tumour growth in both cell lines that had a molecular aberration with FGFR in the KMS11 and KG1A, but not in CALO6 that was wild type for FGFR. And we did have a positive control in that study that led to an impact on tumour growth. So it was a really nice correlation in the in vitro data leading to the same effect in vivo, and we were also able to show the same pharmacodynamic and target engagement effects in these models in vivo. As this project progressed, the disease area changed and a focus on lung cancer was emerging. FGFR1 amplification was shown to be amplified in lung cancer. If you look at the left-hand side of the slide, this waterfall plot shows various lung cancer cell lines and sensitivity to the FGFR inhibitor. And 2 cell lines were exquisitely sensitive to FGFR1 in terms of proliferation effects. And both of these were FGFR1 amplified. And we were also able to show effects on signalling downstream of FGFR1. Unfortunately, both of these cell lines didn't grow in vivo. So in order to test that hypothesis in vivo, we moved to look at patient-derived explant models. So the middle panel, which shows the immunohistochemistry, shows 6 models that were selected based on their FGFR1 amplification status and also FGFR1 protein levels. So you can see the FGFR1 amplification assessed by FISH and IHC looking at protein levels. So we had varying levels of amplification and protein expression, and we had one model that was selected that had normal FGFR1 and was negative for FGFR1 protein. And if you look at the right-hand side, you can see the tumour growth inhibition curves for those models that were treated with 4547, and varying levels of efficacy was observed from 60% tumour growth inhibition through to regressions on those models that had amplified FGFR1. And the model, which was the LCO36, which wasn't amplified, no efficacy was observed. So amplified FGFR1 models were highly sensitive to 4547, and this data helped move the compound forward into clinical trials in lung cancer, and it's still currently in phase 2 clinical trials. Okay, so the next case study is looking at AZD8835, efficacy and mechanism of action, and this is using SYNGENEIC models, which I mentioned earlier. So this pathway is frequently activated in cancer and promotes tumour cell proliferation and survival. And there are many inhibitors in clinical trials, mainly as tumour targeting agents. 8835 was developed to treat solid tumours with activating mutations in the PI3 kinase pathway. And really the impact on tumour microenvironment and anti-tumour immunity wasn't really clear. So we know that some of these PI3 isoforms can impact immune cell function. So what we were trying to do was actually take this molecule and can almost revisit the mechanism using SYNGENEIC models. So exploring the effect of 8835, which inhibits alpha and delta PI3 kinase on the tumour immune microenvironment using SYNGENEIC models. So if we look at the box on the left-hand side, we took CT26, which is a SYNGENEIC model. So it's a murine cell line that's grown in an immune competent animal. Along the top in the first slide is the individual animal tumour volume data over time. And you can see in the blue is AZD8835, which is an alpha delta inhibitor, versus an agent that is only a delta inhibitor. And you can see that both led to significant tumour growth inhibition in this model. We were confident that this was mediated by immune cells, because if we grew the cell line in nude mice, which lack components of the immune system, we don't see any efficacy. So it gives us confidence it's not a direct effect on the tumour cells, but is in fact immune mediated. So in order to explore the mechanism further, we were able to take the tumours at after treatment with each of these agents and profile them by flow cytometry, so we could explore the infiltrating lymphocyte within the tumour microenvironment. On the right-hand side is some of the flow cytometry data from this publication, where we can see remodelling of the tumour microenvironment in contrast to both agents. So what we've seen was, we've seen a decrease in Tregs, which are an immunosuppressive cell type, and an increase in cytotoxic CD8 positive T cells. Along the bottom are impact on some myeloid cell types, and we see changes in response to alpha delta and not the delta inhibitor. So by profiling in the SYNGENEIC models, we were able to explore a different hypothesis for 8835, showing the interaction with the immune system, and validate 8835 as a novel immuno-oncology drug that's independent of its effects on tumour cells. So lastly, to summarise, target validation in vivo is, you have limited opportunity to test your molecule, so you really want to give it the best chance of success. So you want to have a really good tool compound and choose the right model for the scientific question that you're asking. Understand the PK of your molecule, both acute and chronically, and ensure that you've identified your PD biomarker and establish your PK-PD efficacy relationship. From both of the examples, I've showed that successful proof of concept preclinically for 4547 and has led to lung cancer clinical trials, and so that clinical proof of concept is ongoing. And for 8835, exploring that mechanism of action, a different hypothesis revealed a different mechanism to deliver anti-tumour immune activity for this class of inhibitors. So lastly, thank you for listening, and here is some contact information. Okay, so I'll just have a look at some questions. Okay. Let me see. Yeah, so one of them is, how easy is it to obtain patient-derived tissue for xenograft studies? So you can get patient tissue, and there is obviously biobanks available where you can get some patient samples. The challenge for PDX, patient-derived models, is that the tissue has to be implanted. I think it's within at least 40 hours of resection from the patient, so it has to be fresh. We actually work with a number of CROs who specialise in PDX models. They have centres all over the world where they collect samples, have large banks, and importantly, large banks of well characterised and clinically relevant models. And you can interrogate their database for targets to select your target and identify models that you can test your hypothesis. So, I mean, there is a demand obviously for patient tissue, but it is available. But the challenge with PDXs is getting fresh tissue. Okay, another one. Given the pleiotropic effects of FGF and potential for mechanistic toxicity, where you're limited to dosing locally, and is this the intended route of administration for dosing in humans? So yes, the compound is being dosed orally, and that's how we dosed it preclinically. Unfortunately, there are toxicities associated with FGFR inhibitors. In fact, the first FGFR inhibitor was actually approved last year by the FDA. And interestingly, one of the toxicities that we actually see with these molecules is hyperphosphatemia, which we think is linked with FGF23 signaling, and that's actually used as a non-target biomarker in the clinic for these compounds. So yes, we were able to dose orally, and we believe we had enough of a therapeutic margin to take that forward. And lastly, do SYNGENEIC models have to be used for all immune oncology agents? Are there issues if you have antibodies or ADCs? That's, so I guess no. So SYNGENEIC models are obviously murine cell lines in an immune competent host. If you have a human antibody, then and you don't have a mouse surrogate, then these models are not suitable. I mentioned earlier humanized models, which are an important tool that's under development, where you engraft human immune cells into immune compromised mice and allow the cells to engraft. This then allows you to then grow human tumour cell lines or PDX models onto these animals, and then you can test proof of concept where you have human immune cells and human tumour cells. There is challenges with these models that not all immune populations engraft, can take a lot of time to generate, and there are several immune deficient strains that have been developed that express different human cytokines to enable engraftment of different populations. So I think that kind of highlights the challenge that no one model fits all, and I think it's about what question you're trying to ask of that model and making sure that you're selecting the right model for your therapeutic and your disease indication.
Thanks very much, Lorraine. That's great. Thank you very much. So next time we'll be asking, is my compound safe? And we have 3 talks. Firstly, Richard Knight from Apconics, who'll be talking about making safety part of drug design. And then Pauline Garner from Sequani, how to get your molecules into humans. And finally, Malcolm Hattrick here from Medicines Discovery Catapult, with challenges and opportunities of complex cell models in toxicity testing. So I look forward to seeing you all again next week. Thanks again to the speakers, and thank you all for listening.